Good morning, my dear students. Welcome to our zoology online class. Hope you all studied the last class portion. Okay, so in the last class, we studied about the digestion in buccal cavity and digestion in the stomach. Okay, so already I told you maximum the food materials are digested inside the buccal cavity, and some amount of food materials are digested inside the stomach, and the maximum, okay, the major food molecules are digested inside the small intestine. Okay, so buccal cavity, stomach and small intestine. These three areas are acting as the site for the digestion. Okay, so in these areas only maximum food materials are digested. Okay, so hope you are studied about this digestion in buccal cavity and digestion in stomach. Okay, dear friends, today we are going to study about one very important 5-mark question: digestion in small intestine. Okay, digestion small is a very big question and it is very important 5-mark question. And in this topic, we are going to study about some important enzymes and their functions. Okay, so many enzymes are involved in the digestion in small intestine. Okay, so now we are going to study about all the names of the enzymes and how these enzymes are doing their functions inside the small intestine. Okay, so dear students, listen very carefully. Again, I am telling you this is very important 5-mark question. Okay, so digestion in small intestine. Actually, the bile is secreted by the liver. Okay, bile is secreted by the liver. Then pancreatic juice is secreted by the pancreas, and the intestinal juice is secreted by the intestine. Okay, so bile, pancreatic juice, and intestinal juice. These three secretions are directly released inside the small intestine. Okay, directly released inside the small intestine. So the substance present in the bile, pancreatic juice and intestinal juice only helps the digestion process. Okay, so these substance present in these secretions help us to easy digestion. Understand of you? Okay, so dear friends, the next point about this digestion small intestine is same. Already we have studied the wall of the alimentary canal. Okay, is made up of 4 layers. Okay, so here muscularis is the second layer. The first layer is called serosa layer, second layer is called muscularis, third layer is called submucosal, and the inner layer is called mucous membrane. Okay, so these are the 4 important membranes or layers present in the wall of the alimentary canal. Here, the muscularis layer playing a very important role in the digestion in small intestine, because the movement of this muscularis layer is very important to mix the food materials with the substance. Okay, so the food materials are mixed with the bile, food materials are mixed with the pancreatic juice, food materials are mixed with this intestinal juice because of the movement of the muscularis layer of the small intestine. Understand, like you understand? Finally, this movement facilitate or increase the absorption process or digestion process, not absorption, digestion process. Understand, like you? So, dear students, now we have completed 2 points about this topic, and the third point, say, already I told you the bile, pancreatic juice, intestinal juice. These 3 substances are directly released inside the small intestine. Okay, so now we are going to study about the components and the enzymes of the bile. Okay, so the bile contains many components. For example, bile pigments, salt, cholesterol, and phospholipids. So these are the some of the components present in the bile. Okay, bile pigments, bile salt, cholesterol, and phospholipids. These are the substances present in the bile. Here there are 2 types of bile pigments are present in the bile. There are 2 types of pigments are present in the bile. One is called bilirubin and the second one is called biliverdin. These 2 are produced when the hemoglobin molecules are destroyed in the aged RBC. Actually, the lifespan of the RBC, red blood cells, is maximum 120 days. Okay, so after 120 days, automatically this RBC is break down. When this RBC is break down, the hemoglobin molecules are also automatically broken. So these hemoglobin molecules are releasing these 2 pigments. Okay, so the bilirubin and biliverdin, these are the bile pigments present in the bile, but they are the product of the breakdown of hemoglobin molecule. Okay, they are the breakdown product of the hemoglobin molecule. Understand of you? So bile pigment, bile salt, cholesterol, and phospholipids are the substances present in the bile. So hope you are understood this point. Then next point. See the functions of this bile salt. Okay, Okay, bile salt convert the large fat molecules into small fat globules. Okay, so the large fat molecules present inside the ingested food materials is broken or converted into small fat globules with the help of the bile salt. Okay, and second function of this bile is the bile activate the lipase. Lipase is one of the very important enzyme. Okay, so lipase is activated by the bile, and because this lipase is used to digest the lipid proteins. Okay, it's used to digest the lipid protein. Understand of you? So here one more question is there. The enzyme lipase is activated by dash. Okay, bile only activate the lipase enzyme. Understand of you? So dear friends, now we have completed about the bile. Then next we are going to study about the second substance, pancreatic juice. Okay, pancreatic juice. Okay, like bile, the pancreatic juice also consists of many substances and many enzymes. For example, citrullinogen, chymotrypsinogen, carboxypeptidase, pancreatic amylase, pancreatic lipase, and nuclease. These are the some of the very important enzymes present in the pancreatic juice. Okay, so once I repeat all the enzymes present in this pancreatic juice: trypsinogen, chymotrypsinogen, carboxypeptidase, pancreatic amylase, pancreatic lipase, and nuclease. So these are the enzymes of the pancreatic juice. Understand of you? Understand? Then the intestinal mucosa secreting one very important enzyme. Name of that enzyme is called enterokinase. Okay, this enterokinase enzyme only converting the trypsinogen into trypsin. Okay, actually this trypsinogen is in inactive condition. Okay, it's a precursor enzyme. This trypsinogen is converted into trypsin with the help of this enterokinase, and this enterokinase is secreted by the intestinal mucosa. Understand a few? Then next, the trypsin. Functions of this trypsin. Trypsin convert the protein into polypeptides and peptides. The functions of the enzyme trypsin is converting the proteins into polypeptide and peptides. Understand a few? Understand? Then chymotrypsin is very important enzyme present in this pancreatic juice. This chymotrypsin hydrolyzes the peptide bonds associated with the amino acid molecules. Okay, so amino acid molecules are connected with each other by a bond. This bond name is called peptide bond. Okay, so this peptide bond present in this amino acid molecules are hydrolyzed by what can say chymotrypsin. Chymotrypsin. Understand my dear friends? So now we completed in the pancreatic juice. Now we completed the enterokinase enzyme, trypsin enzyme, and the chymotrypsin enzyme's functions. Okay, then now we are going to study about the remaining enzymes. Say pancreatic amylase. The functions of this pancreatic amylase is converting the glycogen into starch and maltose. Converting the glycogen into starch and maltose. Dear students, already told you in this topic we are going to study about many enzymes and functions of the enzyme. Okay, no other points are there. Okay, so this glycogen is converted into starch and maltose with the help of the enzyme pancreatic amylase. This pancreatic amylase is also present in the pancreatic juice. Okay, then next, lipase. Lipase also present in the pancreatic juice. So this lipase converting the fat molecules into fatty acid and monoglyceride. Okay, so the fat molecules are converted into fatty acid and monoglyceride with the help of the enzyme lipase. With the help of the enzyme lipase. Okay, then these monoglycerides are again hydrolyzed into fatty acid and glycerol. Fatty acid and glycerol. Hope you understood this point. Here the fat molecules are converted into free fatty acid and monoglycerides. Then the monoglycerides are again hydrolyzed into fatty acid and glycerol. Understand a few? Then next enzyme is called nuclease. Next enzyme is called nuclease. So the nuclease is the enzyme present in the pancreatic juice. This enzyme converting the nucleic acid into nucleotides and nucleosides. Nucleic acids are converted into nucleotides and nucleosides with the help of this enzyme nuclease. Understand, dear friends? Then the next important point is the secretions of the Burners gland and the intestinal glands. Okay, the secretion. Okay, the fluids secreted from the Burners glands and the intestinal glands are called succus entericus. Understand my dear students? So dear students, now we have completed the enzymes present in the pancreatic juice. Okay. So the enzyme and the substance present in the bile already we have completed, and now we have completed the enzymes present in the pancreatic juice. Understand that you? Understand? Then now we are going to study about some more enzymes names. Okay. See for example maltase, sucrase, lactase, then peptidase, nucleotidase, nucleosidase, lipase. Okay, like that many enzymes functions now we are going to study. Listen very carefully, and this is very important for 1 mark question also. Okay, dear students. Okay, the first one, maltose. Okay, maltose is converted into glucose and glucose, converted into two glucose molecules with help of an enzyme called maltase. Okay, so maltase is the enzyme used to convert the maltose into glucose and glucose. And next, sucrose is converted into glucose and fructose with the help of the enzyme is called sucrase. Okay, so sucrase enzyme is used to convert the sucrose into glucose and fructose. Then next enzyme is called lactase. This lactase enzyme converts the lactose into glucose and galactose. Okay, so lactose is converted into glucose and galactose with the help of the enzyme called lactase. The help of the enzyme called lactase. Understand? Then next, dipeptides and tripeptides. So these two peptides are converted into amino acid with the help of that one enzyme. Name of that enzyme is called peptidase. So peptidase enzyme's function is converting dipeptide and tripeptides into amino acids. Then next, nucleotidase. Next enzyme name is called nucleotidase. This nucleotidase convert the nucleotide into phosphoric acid and nucleoside. Okay, nucleotidase is convert the nucleotide into phosphoric acid and nucleoside. Understand of you? Then next enzyme is nucleosidase. Nucleosidase. This enzyme convert the nucleoside into sugar and nitrogen base. So the sugar molecules are nucleoside molecules are converted into sugar and nitrogen base with the help of nucleosidase enzyme. Understand? Then next, the last enzyme is lipase. Lipase enzyme convert the diglyceride and monoglyceride into fatty acid and glycerol. Okay, so the diglyceride and monoglycerides are converted into fatty acid and glycerol with the help of the enzyme lipase. Understand, my dear students? So now we have studied about different types of enzyme. Okay, many enzymes names now I told you, and I explained the functions of all these enzymes. Understand of you? Understand? Then next point, the mucus and the bicarbonate present in the intestinal juice is providing the alkaline medium for the activity of enzymes. Okay, so for the effective functions of the enzymes, that enzymes need a alkaline medium. That alkaline medium is given by the mucus and bicarbonate present in the intestinal juice. Understand, my dear students? Understand? Then the next point is at the end of the digestion process, all the macromolecules, okay, large sized molecules are converted into simple molecules or very small molecules. Okay, large sized macromolecule or the macromolecules are converted into simple molecules. These simple molecules will be absorbed by the cells. Understand? For example, carbohydrates. These carbohydrates are converted into monosaccharides. Carbohydrates are converted into monosaccharides. Proteins are converted into amino acids. Proteins are converted into, and so protein is an example for the large molecule. This large molecule is converted into very small molecules. Name of this small molecule is called amino acid. Okay. Then next example, lipid. Okay, lipids are converted into fatty acid and glycerol. Okay, like that all the large sized molecule, big sized molecule or the macromolecules are converted into very simple or small molecules. Then only the organs can easily absorb their food materials. Understand a few? So hope you all understood all these points. Okay, then see all the simple substances. Okay, at the end of the digestion process, what are the simple substances present in the small intestine? Every simple substance are absorbed by the jejunum and ileum region of the small intestine. Okay. Okay, so maximum the absorption processes takes place in the jejunum and ileum region of the small intestine. You all know that the small intestine is divided into three regions. First region is called U-shaped duodenum, the next one is called jejunum, and the third region is called ileum. Okay, so here the simple food substances are absorbed inside the jejunum and ileum region of the small intestine. So hope you are understood this point also. Okay, then the next point is some hormones and nerves also controlling the coordination between different organs of our digestive system. You all know our digestive system is a well developed complex system. It is made up of large number of organs. Okay, but there is a coordination is present in between the organs of our digestive system. This coordination is controlled or regulated by some nerves and hormones. Nerves and hormones. Understand of you? Moreover, some local hormones also produced inside the intestine. Okay, local hormones also produced inside this inside the intestine, and these hormones also controlling the secretions of the digestive system. Okay, so local hormones also produced inside the digestive system. These hormones controlling the secretion of the digestive juice. Understand of you? Understand? So dear students, now we have completed one very important 5-mark question, digestion in small intestine. Many 1-mark questions are there. Okay, many 1-mark questions are there, and many enzymes names and their functions also there. Okay, so study today itself, try to complete these questions. Again I am telling you, it's a very important 5-mark question, digestion in small intestine. Okay, so we will meet in the next class. Thank you.